Coming up on Dr. Kiki Science Hour, we're talking about the stem cell madness with Dr. William Gunn from Mendeley. That's up next on Dr. Kiki Science Hour.
Netcasts you love.
From people you trust.
This is Twit.
Bandwidth for Dr. Kiki's Science Hour is provided by Cachefly at CACHEFLY.com.
This is Dr. Kiki's Science Hour, episode 61, recorded on Thursday, August 26th, 2010. Injunction, Junction, what's your function? This episode of Dr. Kiki's Science Hour is brought to you by Carbonite. Backing up the files on your PC or Mac is safe and easy with Carbonite. For a free trial plus 2 free months with purchase, go to carbonite.com, offer code Kiki. Welcome, everyone, to another episode of Dr. Kiki's Science Hour. I'm Dr. Kiki, and during the next hour, yes, we will talk about science. It's what you come to know and love from this show, right? This week, we're going to be getting down and dirty into the science of injunctions. Injunctions? Isn't that a political term? What are we talking about injunctions for on a science show? Well, this week, the big news is that a federal judge has made an injunction, created an injunction against government stem cell funding. So, what is happening in the world of stem cell funding? How is this going to affect stem cell research, specifically embryonic stem cell research, which is what the injunction is specifically for? And let's talk about the research itself. Let's talk about some history. I want to get down and dirty into this topic that, for some people, is incredibly emotion-ridden. This is a very political subject, and I believe that this injunction has created, a scientific quagmire that reaches from the laboratory all the way to Washington. So to join us today, I am joined by Dr. William Gunn. He is a assay development consultant, and he's also the community liaison for Mendeley. He is self-employed out of San Diego, California. He's a biological researcher. Without further ado, say hello to Dr. William Gunn. Thanks for joining me.
Thank you, Dr. Kiki.
Yeah, it's really good to have you here today. And you have actually, the reason why you're able to talk about this subject is you actually have personal research experience in the stem cell arena. Can you give us a little bit of your personal history and what this injunction kind of, just makes you start thinking of?
Well, I started my PhD research in a stem cell lab at Tulane University, and our lab focused on adult stem cells, not the embryonic stem cells that are the focus of this, but we kind of got drawn into it a little bit as one of the arguments the anti-ESC side made often was that, well, we have these other kinds of cells that people could use. We don't need to use ESCs. And of course, the science doesn't support that at all. But I did get a couple years of being immersed in these issues, and in fact, the plaintiffs actually in this case, the ones that they finally found that had standing to bring this suit, are adult stem cell researchers who are supposedly being harmed because they might have to now compete for funding and share a smaller piece of the pie with a lot more researchers. That was how their standing in the case was determined.
Right. So. Yeah, and that's a really, I think, just a really interesting point to make is the way this court was at this case was actually brought to court is they had their proof of injury was that they study adult stem cells. And they said, but we compete for funding against this. I mean, what if these embryonic people get money and we don't get money? That's just not fair. you know, and
Yeah.
the legal system decided that that was a fair proof of injury. And I'm just wondering what kind of precedent this sets for just anybody, you know, suing the NIH just because they're competing for funding.
Yeah, it sets a really scary precedent because unless they increase the amount of funding for research to the NIH a whole lot, pretty much every policy decision that touches the NIH budget is going to create a standing for someone to sue about it if this precedent holds. So it's really really an unfortunate decision that I think is going to have really wide-reaching ramifications beyond embryonic stem cells or stem cells in general. It's pretty far-reaching, and the only cure really is to remove the act that started all this back in the Clinton era, the amusingly named Dickey-Wicker Act Amendment.
Right. Do you know much about the Dickey-Wicker Amendment? Can you talk about what that's done just in general to stem cell funding?
Well, what it did initially was to mandate that federal funds couldn't be used in any case where an embryo was destroyed. Now, during the Bush era, the policy was that there was a distinction between the research that was done and the destruction of the embryo. So for a long time, people were able to continue doing research with stem cell lines that were already established and with cells that they were able to obtain otherwise. But this recent decision kind of reaches beyond all of that. So, we're going, in a sense, a step back even further than we were during the Bush era.
Yeah, that to me is just amazing. I mean, the fact that with this one injunction, it's a sweeping ban for funding that it takes it not even to the level of the existing stem cell lines, which was what the Bush era mandates allowed, but to even prior to that, which is just nothing. Right? No, and the wording, what it's based on, wasn't it based on a reinterpretation of the word research?
Yeah, they sort of parsed this a little bit finely, and what they said. I actually have a quote over here somewhere from one of these guys, where they give the exact sort of way that they dice these words together. So the distinction between the process of creating the cells and the research performed on the resulting cell lines. So since all of these cell lines had been created through this process that destroyed the embryos, but they were already existing and cell lines weren't being continually destroyed, or embryos weren't being continually destroyed for this, that was what it was allowed. So it was. Now what they've said is that research on embryo-derived stem cells is no different than research that destroys the embryo itself. And that was really the crux of this ruling.
Yeah, it's such an interesting parsing, that the fact that in research, which we use the term research as a general term, but then there's research to specifically delineate one line of research. There is research to delineate a particular study. In academic papers, you see these multiple uses of the word research over and over again, and yet the judge based in being able to make this ruling and say, okay, so now there's absolutely no difference. He's basing it on this one use of research that comes from the Merriam-Webster dictionary that doesn't even it isn't even indicative of how it's been used in law, in politics, in science for over the last 10 years.
Yeah, I think he had a good point when he said that if the people who wrote the law had wanted it to be interpreted in these two different ways, they could have written that into the law. That was a good point that he's making, but only the really beneficial outcome we could get at this point is if this is sort of the point at which things get bad enough that we realize that, okay, this halfway solution we've had really isn't working for us at all, and we get a real and lasting solution to this that goes a little more broad.
Yeah, I think that's going to be the interesting, where it's going to get interesting is can anybody from this? It's going to have to go from here. Where's it going to have to go? We're going to have to. This judge is trying to make a very broad statement. The plaintiffs were trying to make a very broad statement that will just have sweeping effects. But now it can be appealed to the Supreme Court, right?
It's being appealed, yeah.
Yeah, so it can be appealed. It's going to be appealed to the Supreme Court, but also in the meantime, this is going to get punted to the election season. And so this couldn't this just become an election season issue where, candidates are running based on whether or not they agree with embryonic stem cell research funding?
And does that remind you of any election in the past? I don't know. 3 or 4 election cycles? Yeah, pretty much. So I think this is all part of the plan.
Yeah, it definitely is part of the plan. It just is it just continues to boggle my mind. I'm like, aren't we done with this yet? Haven't we just put this issue to bed yet? No, obviously we haven't. To kind of go backwards a little bit. I mean, embryonic stem cell. I mean, what exactly is an embryonic stem cell?
So the embryonic stem cell is an unfortunately named construct. It is cells that were derived from about 4 to 8 morula or blastocysts. So we're only a few cell divisions from a fertilized egg. And these cells, at this stage, retain the ability to produce an entire organism. The reason they're named embryonic stem cells is sort of in parallel to an earlier system that researchers studied, which was the embryonal carcinoma cells. So researchers back in the 70s and 80s had found this interesting phenomena where certain cell masses called teratocarcinomas could form a variety of different cell types within the mass. You could find bone and epithelium and little bits of other organs in there. And so this sounds kind of gross, but to a researcher this was fascinating because they realized this is a potential to study developmental processes with a view really towards regenerating these sort of organs in vitro. So if these could be harnessed, they thought one might be able to grow new and replacement organs instead of having to put people on these years-long transplant waiting lists. And engineering these things to be more immunologically compatible could extend the lifespan of people. They wouldn't have to be on such heavy regimens of immunosuppression drugs and so on and so forth. But to get back to the original idea, they had these embryonal carcinoma cells that they were studying. They're derived from these teratocarcinomas, and they call them EC cells. When they found these other cells derived from the embryo were a lot like these embryonal carcinoma cells, they kind of named them in parallel embryonic stem cells as opposed to embryonal carcinoma cells. So it made logical sense from a research point of view. But boy, I wish they had called them, morula-derived stem cells or blastocytic stem cells or something. It was just one of those mistakes, and you never really can predict what the consequences are going to be. And science has made these mistakes over and over again.
Many times. Oh my goodness.
Yeah. So.
Sometimes I can't. The foresight of scientists, unfortunately, was not such that they thought about the reactions to the word embryonic that contains embryo. That's something that people are going to they have an image of that in their head, and they're going to respond to that in an emotional way. The foresight, if they had just called them morula stem cells or blastocytic stem cells, it just wouldn't have been as catchy. It wouldn't have been, who's gonna grab onto that? People probably never would have. It would have been a non-issue. Yeah,
it would have been something else we were arguing about. We have all kinds of things that science touches on that are, really raising the larger question of how much PR do scientists need to be doing versus, how much do they need to be left alone to do the research that we're paying them to do. So that's fights are ahead over this issue, and we don't have to get too far afield into that on this call, but it is a fascinating topic that's getting more and more awareness these days.
Yeah, which is great. I'm glad that it's getting more awareness, and I'm sure the up-and-coming generation of scientists are probably getting more and better training in media and becoming more media savvy, and also just being part of the internet generation. You're more into being connected and like what's happening on the web in general. So there's probably a large part of that that's going to influence future science and future terminology choices. But it's just it's interesting to me that and who would have the foresight? And wouldn't it have been nice if someone had had the foresight differently? Would it have changed this discussion to something entirely different? And
Yeah, I'm just waiting for the people who study Drosophila to have their day, naming their genes weird things like, Sonic Hedgehog and Son of Sevenless, and, making obscure references to Lou Reed songs in their gene names, that kind of thing. One day their day will come.
Yeah, the Drosophila people, so far, they've had it pretty easy. No real...
Maybe that's the secret. Maybe pick something really obscure and then people can't, catch on to it.
Maybe. I don't know. I know Sonic the Hedgehog, the media caught on to that just because of the name, the Sonic Hedgehog gene. It's like, really? When that came up, I remember all the media titles were all about, video games and comparing, and
Right.
ridiculous.
Yeah, silly stuff.
But in terms of getting back to the discussion of what differentiates embryonic research from adult stem cell research? What is an embryonic stem cell? We've gotten through what is an embryonic stem cell, but when we get to the point of in the laboratory, so your personal experience, you worked on adult stem cells.
That's right.
Not on the embryonic. So
that's right.
Your experience was probably very different than what's currently happening or what happened even, years ago to people wanting to work on embryonic stem cells. But, what are some of the things that really differentiate, how the funding goes to one or the other?
Well, as it turns out, the adult stem cells are the ones that are the closest to actual clinical application. There's hundreds of clinical trials going on using adult bone marrow derived and other stem cells for a variety of reasons and applications. There's really only one case in which it's a Geron trial that's using the embryonic stem cells because of this potential that I mentioned earlier for them to create any sort of tissue that's a little bit harder to harness. But you often hear people say, well, we don't have to have embryonic stem cells because we have these, and that's manifestly not true. The science behind mesenchymal stem cells, or the merostromal cells, or any of the other cell types that kind of go under the category of MSCs, is that these are more mature. They're aged cells. They have epigenetic modifications that have happened as they've divided many, many more times. We're not talking about, the 2 or 3 divisions away from the fertilized egg. We're talking about a grown human being now. So all of that patterning and differentiation that's going on in the years is present in these. So they're just not as... So what holds them back from having all of this potential makes them easier to target for a clinical application, but at the same time, it restricts a lot of the things that they can do. So that's where we are with that. I think that the embryonic stem cells are the variants that we have nowadays, which are the induced pluripotent stem cells, which is a way of taking a cell from an adult and converting it to an embryonic stem cell-like phenotype. So it resembles these cells, but it still has some of this, what they call epigenetic imprinting, some of these things that have happened to the cell over the course of its existence in the organism that makes it distinct from the true embryonic stem cell. So we have a variety of different things, but it's sort of a false dichotomy if you ask, well, should we use this one or that one? Because if we want the research to progress, we need to be working at all angles simultaneously because they all have their different applications.
Right. I've been listening to interviews and looking at articles all week long on this subject since the injection occurred, and the arguments seem to be the same things. Well, okay, who cares if they're not funding embryonic stem cell research for reason number one, that we have adult stem cell research being funded, and adult stem cell research is, like you said, making great strides. So, for example, someone brought up the example that a liver had been created from adult stem cells. And I'm like, hey, that's great. A liver, that's awesome. And it's probably a liver that can be matched to an individual because you can take their cells and like use them, induce them to be liver stem cells and create a liver for a person to transplant into it. That's great. But at the same time, I'm not seeing the organs that they're talking about replicating or creating from adult stem cells don't seem to be like the really complex organs. It doesn't seem to be the ones that would be really hard to make.
Right. Yeah. Nobody's making brains in the culture dish just yet. I think that the ESLs have a lot of potential even beyond that. You can, if you can induce these guys from, say, a patient who has a certain disease, then you'll have basically a set of cells that have a certain genetic defect, and you can test these guys in the culture dish for drug screening and creating new therapies. And there's a lot of research that has been started along this area. So it's a real promising area of research. We have a lot of potential applications, but what we're missing in this whole equation is the blue sky potential of the embryonic stem cells. And you never know where something like this can lead. Being too applied can, and focusing on all your research into what's close to being applied can be dangerous. I mean, one of the most striking scientific discoveries of this decade, well, it wasn't discovered this decade, but one of the most striking scientific discoveries is that of the transposons. These came about from a woman, Barbara McClintock, who was studying maize. She was studying, genetics and patterning in this research strain of corn, and a totally, sort of pure research, basic research sort of thing. And what we've come to learn now is that these transposons are not only present in humans, but they're responsible for a lot of the diseases that we face that we hadn't been able to find genes and narrow it down to. So these sort of this sort of research and these sort of discoveries come from unexpected places, and that's what I think the real tragedy would be if we went with that line of argument and said, oh well, we can do what we need to do with these other cells.
Right, because the truth is we don't really know what we're going to need. We don't know where the information is really going to come from. And that's one of the other points there that you've made is that that people have said, we can get rid of the embryonic stem cell research because the adult stem cell research is there, but you mentioned earlier the point that within embryonic stem cell research there can be transfer of information. So even if the applications don't come from embryonic stem cell research, the information about how processes work, can't those initial understandings come from the embryonic stem cell research and potentially not be able to be gathered from adult stem cells?
Well, sure. One of the foremost researchers in this field, Rudy Yanish, the guy who was behind the Dolly the sheep and a lot of this really fascinating work on how genomes are imprinted and how development occurs. He started his work looking at the differences between unimprinted early cells and these more mature cells and what has happened to them later on in their developmental cycle. So it's really kind of an interesting, unexpected thing that you get when you have the ability to look at a whole bunch of different things, make the comparisons between them within this area. So, I mean, we can do in vitro studies with the cells. We can take them from patients with different genetic disorders and look at those. We can differentiate them down different developmental pathways and look at what genes get turned on. All of these sort of things we can do at different cell types, and that yields valuable information that we need to know if we want to reach the goals of regenerative medicine, if we want to actually realize this goal someday of being able to replace an ailing heart instead of just, trust it like some of the things we can do nowadays.
Right. I'd like to take this moment as we're talking about stem cells to move into thanking someone who's good for backing up information, the way stem cells back up information about who we are and how we should be built. Anyway, our sponsor for today, this episode is brought to you by Carbonite.com, the leader in online backup. Unexpected computer disasters happen all the time, and wouldn't it be wonderful if you had everything all backed up? But most people don't have everything backed up because, it's too hard. I don't have an external drive. All these excuses, right? Some of the common problems are hard drive failures and viruses. I just recently had that happen personally. Hard drive, kaput. Got a lot of information that's just gone. And the result? Like I said, just gone. We risk losing the files on our computers. So I recommend backing up with Carbonite. I wish I had, actually. I really do wish I had. It's safe, it's easy, and it is an automatic backup for your PC or your Mac, both platforms. Carbonite helps protect all of the valuable files on your computer, your photos, videos, music, Word, Excel files, financial documents, PDFs, all that stuff that's important to you that when it's gone, you go, oh man, where did it go? A few more things you might want to know about Carbonite before you buy into it. It's safe, right? It's very safe. Your files are encrypted, so they're not going to be magically hacked on the way to the storage site. They're encrypted before they leave your computer for maximum security. It's easy to use, whether you're a computer novice or, super techie of the world. It's automatic. You don't have to remember to do it, which is the crucial thing here. You set it up, it just hooks in, backs up your files automatically whenever your computer is connected to the internet. So it's no longer a problem of yours. It's just taken care of. You get your files back in a really easy number of clicks. So if a problem does happen, you have to replace a drive, it's just a couple of clicks. And because your files are backed up off-site, you can access these files from any computer, your phone, your BlackBerry with their free apps. And if you back it up, you can get it back from wherever you are with Carbonite. So here's the offer, everybody. If you, like me, need to back up your precious information, you can get unlimited backup for your PC or your Mac for only $55 a year. That's pretty reasonable when you think about the price of some drives, right? This is a pretty reasonable deal. That's 15 cents a day. It's less than your daily cup of coffee. It's really, really, really not so bad. Try it for free. Free, what? That's right. Try it for free. That's even less than 15 cents a day. You just have to go to Carbonite.com, enter the offer code Kiki, K-I-K-I. That's right. Go to Carbonite.com and enter the offer code Kiki. If you decide to buy the service after your trial period is over, of course, you'll get 2 months free. That's still free. Wait, you had a free trial and now you're getting 2 months free with your offer code of Kiki. That's right. Because they know people like me need help backing things up. So be sure to sign up for the free trial from the homepage to get the 2 months for free. You don't have to have a credit card for the free trial. And remember, once again, it's Carbonite.com, offer code Kiki, to back up all that stuff that's important to you. And I'd like to thank Carbonite.com once more for sponsoring this episode of Dr. Kiki's Science Hour. And without any further ado, let's get back to the science. We were talking about stem cells, which really do back up all of the information in our genome. Something goes wrong, the stem cells are there to fix it. You need new skin. Stem cells, they divide and grow, and you have new skin. It's just amazing. But there are problems with stem cell research these days. Unfortunately, it's a battle. It's a war zone out there, really. Researchers who are studying embryonic stem cells are no longer able to get federal funding for their research as a result of a federal court injunction just earlier this week that took place. All of a sudden, money gone. People like Francis Collins, who's the head of the NIH, they're saying it's a mess. It's really a mess, and this is not something that's going to be helping move research forward in this area at all. I mentioned earlier, one argument that I've been hearing about stem cells being, well, we don't need to study embryonic stem cells. We have adult stem cells. But another reason that people argue consistently is that why do we need public funding? Why do we need public funding for embryonic stem cell research? Why not? They can just get all their money from private funds. So, Dr. Gunn, do you have any ideas? I mean, you've had experience with the adult stem cell side of things, but how is the landscape out there for the private versus public funding, and is it really going to make a dent?
90% of our funding came from public sources, from the NIH. We did have a small amount of foundation funding, but it was just nothing in comparison to what we were able to get through the NIH and through public sources. So really, singling out embryonic stem cells and asking why do we need federal funding for this is just part of the larger question of should we be funding, research on whatever, a given interest group likes or doesn't like. If we start making these decisions, we run into those large ramifications that issues like this bring up. And what happened back when they originally passed these, the Dickey-Wicker Amendment and these sort of things that really restricted the research, was that states ended up funding most of the research. At California, you had the California Stem Cell Institute of Regenerative Medicine. They had a big bond issue to pay for that. New Jersey had another similar, $100 million bond issue for strictly for doing research on these kinds of cells. So really, one could argue that by bringing this issue into the public awareness and having this motivation behind it, there was more funding made available for embryonic stem cell research than there had been, just because of all the attention that it was given. But that sort of thing is not really a sustainable way to do research when you consider that the vast majority, again, 90% or more of all labs depend on federal NIH funding for their work. So there's just, research is expensive these days, and we can't go making decisions to do state ballot initiatives just for one little section of research to get around a temporary public squeamishness. I mean, I'm sure there's lots of things that I'm sure that for any given area of research, you could find some group out there that would be opposed to it. It's sort of like, the Rule 43 of research,
Right.
I mean, you can find somebody who's against it. So, research is a public good. There's good things that come out of it. So it should be funded publicly. That's really all there is to it.
Yeah, and I think part of the questions that keep arising are, okay, so people keep asking, what's the benefit? What's the benefit? What's the benefit of the embryonic stem cell research? Why do we have to do it? It's a controversial topic. Why do we have to do it at all? We've already mentioned the fact that discoveries can be made about just the way that cells work, that development occurs. We can learn so much about, ourselves. I keep going back in my head to that old intro biology motto: ontogeny recapitulates phylogeny. And so if anybody who's taken like intro biology, to the animal kingdom has probably heard this phrase: ontogeny recapitulates phylogeny, meaning that the process of development repeats the process of evolution in some senses. And so even looking at the cellular level, I mean, we can probably gain massive insights into what genes evolved at certain points during evolution and why they're important. What do they do? by studying embryonic stem cells, and that we won't necessarily see the same genes activated in the adult stem cells to be able to get that information out. Do you agree?
Right. We one of the areas that we studied in our lab was specific sets of genes that got turned on and off in a timed manner, depending on the stage of maturity of the cells and the stage of development into the tissue they were eventually going to become. There's this sort of orderly pathways. Certain cells, certain genes turn on, do their function, and then they go off and another set gets fired off. And that's the same way cells kind of work in the developing embryo. There's sort of a staged and patterned process for all of this. The whole way that we knew which genes were important to look at for our research, which has been pretty useful for a certain kind of cancer. We've got a treatment for bone-resident cancer called multiple myeloma. It works and it gets its growth signals from this pathway called the Wnt pathway, and that pathway was originally discovered by people who are doing these developmental studies. So we wouldn't have known necessarily where to look among the thousands and thousands of genes involved here had we not had some of this earlier information from developing embryos and in other organisms, So
right.
It's really it's a fundamental thing, and I guess you could say I really think of this more or less in absolutist terms.
Yeah, and I think that's probably where the divide is. People on one side of this argument who don't want embryonic stem cell research to take place, they think about it in absolute terms as well. In terms of some questions that have been brought up, you've worked in a lab where you've received government funding and probably private funding as well to make the research happen. One of the points brought up was that, okay, for this research, for stem cell research, maybe we shouldn't have the people, scientists, who are involved in the research and getting the funding, who would benefit from the funding, involved in arguing this at all.
Yeah, it does.
Isn't there bias involved somewhere?
Yeah, it gives rise to a little bit of a conflict of interest. Objections, and again, that goes back to the basic idea that if we still value research as a society, as a public good, then we have to fund it as a society. There are a whole lot of considerations that go into doing research. When I did my research, not only did we work with stem cells, we worked with human patients, we worked with animal subjects, and there's a whole lot of ethical considerations that you consider first and foremost. And at every university, there's a board that it is not part of your research committee and doesn't stand to gain anything from in terms of grant funding or anything from their decisions, that reviews your experimental plans and makes sure that it's conducted according to proper ethical standards. And these are, there's a consensus about what these standards are that are on a worldwide, nationwide level for the same for animal research as it is for conduct with human patients, and the same thing with the stem cell research. There's no studies of the human embryonic stem cells the way they were doing with animal embryonic stem cells initially, where they would take embryonic stem cells from one morula and put it in another one and see what the resulting organism looked like, creating these things, which they again named kind of, unfortunately, but I think it's strange enough they've gotten a path. They call them chimeras.
Chimeras, I know. It's straight out of Greek mythology.
Yeah. So no one is and no one will be doing any of that sort of research with human embryonic stem cells because we have ethical standards that prohibit that. So it's, there's really a whole lot of self-policing that the research community does about what's acceptable and what makes the most sense. And really the research community is the people who probably in the best position to judge the subtleties and the nuances here. Yes, they are the ones who are getting funded to do the research, and that's their job, but they're the ones that really understand the situation and the nuances and the subtleties of the situation. Like, the whole deal with the embryonic stem cells coming from the morula and just being a few cell divisions away from a fertilized egg. The other point that never gets brought up, that all of these cells in the first place were taken from material that would have been discarded as medical waste the next day had we not done the research on it. So, I mean, there are things that come in here that have ethical weight in the situation that someone who's not familiar with the whole history and how it was set up wouldn't realize. So I do think it is appropriate within science's history of self-regulation for us to be the ones to set our own standards here.
Yeah, and historically, science has, I mean, there have been mistakes made. There have been problems that have been widely publicized, but at the same time, there are, we've made progress. We move past. We learn from our mistakes and we move forward, not to make those mistakes again, and hopefully to become even better in the way that we approach potential problems in the future. What comes to mind from talking about this is the argument of the if they're going to, like, from IVF clinics or excess embryos that are created, excess fertilized eggs that have been created for in vitro fertilization, that if they're going to be thrown out, why not use them for research? Other people have said, there are, hundreds of thousands of people across the country who'd be willing to adopt those fertilized eggs. Very, very, it's probably true. And there are probably some individuals who would take their IVF cast-offs and be willing to have them adopted out. But maybe there are other people who would much rather not and would rather have them go to research. So why not have the choice available?
Well, here's the issue with that. The issue there is viability. Not all of these embryos that are selected for the IVF procedure are going to go on to create a fully developed and normal organism. In fact, they do screening of these things to pick the best one because they've gotten a lot better with it, but there's still 20 or 30% of these that will not ever go on to develop a full-grown organism. So there's nothing to carry to term, there's nothing to adopt. And that's just part of the biology. It's an unfortunate fact that even that everyone who gets pregnant experiences the same thing. There are pregnancies that are not carried to term because something's wrong with the embryo, that are arising out of an IVF clinic. And that's just a natural part of the biology that's programmed in. And these are things you wouldn't want to carry to term, because the reason that the abortion happens, that the miscarriage happens, is because there's something genetically wrong. There's something that's happened that's caused that to that caused the inbuilt sort of safety switch to trip and say, okay, we're going to terminate this procedure because it's things aren't going according to plan. Right.
The organism will not be able to survive outside of the womb on its own. So it would probably something horrendous has gone wrong in the developmental process that would not lead to a viable offspring.
Right.
To break it down to, and it's, you gotta think about it in those ways. It seems to make logical sense, but we can't argue logic sometimes.
It's really very noble that people would want to protect these guys, and that they would want to offer to take the excess fertilized embryos, and that's what I think it's a very noble sentiment. And I think a lot of the people who started asking questions about embryonic stem cell research they really came from the right place in their hearts initially, saying, what are we doing here? Let's understand the ramifications. Unfortunately, things kind of got polarized and it turned into a political football on top of that. So we can't get back to to a reasonable discussion anymore. But still on top of that, you have the fact that it's just a part of nature that you lose some of these guys anyways.
Yep, it is part of nature, and that's something that people need to take more intro biology classes. We need to get this stuff, how things work, how it works, kind of drilled into people, so people at least have a scientific understanding of the process and what exactly is going on. To be able to make informed decisions, however they want to be informed. The information should be given to them.
Absolutely.
Yeah. As we come to the close of the show here, I'm wondering, where, do you have any thoughts on where this is going to end up going? Do you have any hope for public funding? I mean, we'd all love to see the public funding reinstated. Is this just going to be a slog for a long time? Is this are labs just not working right now? Are people just shut down?
Well, it's going to be a slog. So the current state of the situation is that there was $130 million of research that was approved between the time the Obama's policy came into effect and this injunction happened. All of that stuff, the NIH's ruled, can stay. Now, that's just the tip of the iceberg in terms of the grants that were submitted and that were under assessment, but it can't be decided on until this other issue gets worked out. So was a little bit of work done. There's some work that's continuing to be done. We got a little bit of work that was done over these past year or so. Really, really good progress that was made. So I think people are going to be able to look at the good progress that we did make while the Obama policy was in effect, and the work that's done from that was already gotten through before this injunction happened, and see that this really is having a beneficial contribution to the state of research. And I think we will see policy move in the right direction. But it's always going to be a slog. I mean, we haven't in this nation gotten past evolution, which is it's getting to be a centuries, not decades old debate. We haven't gotten past the issue of abortion, which is kind of where a lot of this embryonic stem cell stuff came out of. So I don't think we're going to just magically get over this. I think it's going to continue to be something that we struggle and we deal with, but I'm optimistic that the research is going to continue to prove valuable and that we'll be able to move forward.
Yeah, I absolutely hope so. I'm also optimistic because being in the state of California, this might actually continue to benefit California in the fact that the voters of California have approved a lot of funding that's still being meted out through the California, Institute for Regenerative Medicine, CIRM. And additionally, there are other states who could pass their own state mandates that would not necessarily, so they could give state funding as opposed to having to worry about federal funding. Additionally, there's always the UK. There are always other countries. Researchers can go other places, right?
Well, I'm glad you brought that up, actually, because watching the literature and seeing the amount of work that comes out of places like China and Korea and Singapore, specifically in this area, the most recent Nobel Prize work on iPS cells, that came out of not strictly a U.S. lab, because we've given them this wonderful head start. We've stopped doing the research. Other countries have been able to do it, and you should see all of the publications that come through. They're not from the U.S., and they're the ones that are winning the Nobel Prizes in the future on this. So I think it's really an issue of the remaining relevancy of the U.S. as a research leader.
Does this, I absolutely agree. I wonder just in terms of the regulations and how, I know that in laboratories that get federal funding and private funding, they have to have different equipment that's bought by the federal funded, with federal funding and other with private funding. I'm wondering if somebody is collaborating, somebody with federal funding is collaborating with somebody overseas, is that going to affect their ability to continue to collaborate on a project like this?
Well, you still have to maintain a firewall, literally, between all of the funds that are spent, on that research and all of the other work. And there may be some people who travel back and forth and do this research. I know that, for example, Alan Tronson, that one of the guy from CIRM, he traveled back and forth between Australia for a while. But that's not really a solution for anyone. It's sort of wasteful keeping this firewall in place, and one could argue that it would do a lot better if things were allowed to mingle and work as they should.
All right. Well, thank you. I know we're over our 5 o'clock hour here. We got started a little bit late, but thank you for talking about this subject with me today. I do appreciate your time and your opinions and your experience in the area. It's I think this is it's a fascinating area. Every time something comes out in this area, I'm all over it. I just love it. Unfortunately, it keeps happening.
Yeah.
But thank you so much for being able to discuss this with us today. I do appreciate it.
Thank you very much, Kirsten.
You're welcome. And if anybody is interested in following Dr. William Gunn, you can find him on Twitter. He is Mr. Gunn on Twitter, and additionally, you can find his profile, articles that he has published on Mendeley.com. So his website there, Mendeley.com, MENDLEY, /profiles/William-Gunn.
Yes, and Dr. Kiki, there's one thing I would like to add. I just put together a collection using the Mendeley Public Collection feature, which is going to be listed under my research collections area there for all the MSC literature and the embryonic stem cell literature. So you can find a public collection from me with a lot of the basic research from guys like Tronson and Rudy Yanish on embryonic stem cells and MSCs as well.
Great, and I think that's a great use of Mendeley and the social aspects, being able to open up papers for people to be able to search through and get an eye on. Mendeley is a great. It's not an app. Network.
Yeah.
It's a great network. It really is, for anyone who hasn't checked it out, who's interested in who's in science research. This research network, it's a great thing. You might want to take a look at it. But anyway, we will move on to the end of the show. Thank you once again. Have a great evening, and I will see you on Twitter.
See ya. Bye-bye.
And that's it for Dr. Kiki's Science Hour. Once again, we have spent the hour discussing science. I will be back here next week to talk about more and more science with somebody incredibly interesting. Hopefully, we'll be able to get more interactive stuff set up so I can take your calls at some point. I do like doing that. It's neat to get your phone calls. But in the meantime, you can call my Google Voice number, but I forgot what it is. So I'll put it on Twitter. You can find me on Twitter. You can tweet me questions if you have any questions at any point in the future. I'm Dr. Kiki on Twitter. I am Dr. Kiki on Facebook. My website's drkiki.tv. If you want to download past episodes of this program, go to twit.tv/Kiki. I'm there in audio and video glory. Additionally, there are videos up on YouTube, so if you want to post these videos on your website, you can do that too through their embedding stuff. That's really a great way to share this science with the world. You can take an episode of Dr. Kiki, put it on your website, and everyone who comes to your website will get to see it. It'll be pretty awesome. Also, I will post the URL because I, again, don't remember it. It's a little bit complicated. I'm having a remix contest for my theme song. So anybody who is interested in taking a stab at remixing my theme song in, lots of different, you could make it country western, you could make it electronica, whatever you like. Remix the theme song. There is a website with all of the music files available so that you can actually grab them and remix them the way that you'd like. And I will post that for you. Make sure it's in the show notes so that you can try that if that's what you'd like. I already have one remix that somebody has sent me that's pretty darn good, so I'm excited about that. And that's about it for right now. If this hour of programming did anything for you, I sure hope it made your world a lot more interesting. See you next week. Just want to say thank you to everybody out there for watching the show. Thank you to everybody for all of your all of your nice comments and support since my big announcement today. So I really appreciate that. It's going to be 6 more months of, a little less than 6 months actually. Yeeks! 6 more months of craziness. So anyway, I hope that we grow together, right? It's going to be good. It's going to be fun. Don't worry. I'm, I won't disappear. I won't disappear when I become a mom. I will, I love doing stuff like this. I mean, who knows if I will be doing the Science Hour? Who knows if I'll be doing another show? What exactly I'll be doing? But I'm not going to disappear. I like science too much, and I like communicating science, and I like talking to people about it. So I like teaching. So it's really something, I like asking questions. It's very important to me, and I think without it, I would have a little less interesting, fulfilling of a life. So, I mean, it'll be completely different, so I have no idea what it's going to be like, really. But I'll be around. I will be around.